Hello, everyone, and welcome to today's live broadcast, Differential Abundance Analysis for Microbial Marker Gene Surveys. It is presented by Joseph Paulson, PhD, a research fellow at the Dana-Farber Cancer Institute. I am Judy O'Rourke of LabRoots, and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots. LabRoots is the leading scientific social networking website and producer of educational virtual events and webinars. Before we begin, I'd like to remind everyone this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box that appears on the screen. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, please click on the Help Desk button located in the promotional board at the bottom center of your screen, or use the Ask a Question box to let us know you're having a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located in the top right corner of the presentation window and follow the process to obtain your credits. Please join me in welcoming Dr. Paulson. I will now turn the presentation over to him.
Hi, everyone. Thank you, Judy, for that introduction. So I recently joined Genentech, but everything that I'm going to present today is work that I've done at Dana-Farber or that I've done at the University of Maryland beforehand. And I'm going to talk about differential abundance analysis of marker gene surveys. And really, the overarching theme, it's applicable to not just for microbiome or metagenomic assays, but these are approaches that are sparse aware. They're approaches that can account for the sparsity, for the zeros that are in that count matrix when one's doing an analysis. So I'll jump into it and give a little bit of a background. After sequencing, typically you go ahead, you curate whether you're using QIIME or Mother or one of these tools to go from reads to count matrix for the analysis. This is where you would step in and start using these sparse aware methods. And so the overarching theme really for this talk is about the missing information and technical artifacts present, ubiquitous really, in marker gene surveys, 16S amplicon sequencing, and also ubiquitous in whole metagenomic shotgun sequencing. And there are, as single cell sequencing is coming about, this might be, it's not validated, but it might be applicable, or sparse aware methods and approaches might be applicable as well in that arena. So, but the majority of the talk today is really going to focus in on the microbiome, and in particular on 16S sequencing. I'm going to highlight several old papers actually now, and try to tease the audience with some more recent development that I'm happy to answer questions about and or present at a future in the future. So one more time, really everything is about what's missing. With the Human Microbiome Project, and even before this, as we were starting to look at different microbial communities, when I was first, before the Microbiome Project came out, I was analyzing a big 1,000 patient cohort where we had done 16S sequencing, and we just noticed that there were so many zeros in the matrix. Here I'm plotting the read depth from all the samples, all the 16S samples in the Human Microbiome Project along the x-axis, so that's the sequencing depth, or a proxy for it, the number of reads within the count matrix. And along the y-axis are the number of OTUs, operational taxonomic units, that were detected. And you see this nice linear trend that as you sequence more, deeper, you observe more in general, right? It's not always the case, but there's a strong correlation there that as the technology, this was actually 454, but whether it's Illumina or no, I don't remember which technology this was. I believe it was 454. But as you sequence deeper, more, you get more and more OTUs. You discover more bacteria. Now, if you look at the bottom left corner, right, you see a bunch of samples over by 0, 0. Those particular samples they have very few reads. They have maybe 1, 2, 3, 4, 5 reads, and they also have detected only 1 or 2 or 5 OTUs. You would not imagine that a particular sample, a particular patient, some of these were gut, right? You would not imagine that a gut sample has only 5 bacteria in there. So that in that particular instance, right, you would imagine it's a technical artifact. But as you move along the x-axis and you get into higher sequencing depth, right, you observe more and more bacteria. And so what we want to do, we want to take into account the sequencing and technical artifacts due to the technology, due to either low sequencing depth or high sequencing depth. And we want to develop statistical approaches to account for that and to estimate the probability whether or not a 0 belongs to some sort of technical distribution, that, okay, there was an earthquake and the sequencing machine didn't actually spit out enough information, or that particular run went really well and we expect that all the zeros in the matrix, I'll go over this again in a couple of slides, but all the zeros are real biologically meaningful absent bacteria. So these are the main issues that are really present in microbiome marker gene surveys, at least for me from an analysis standpoint. And it seems to be a trend coming out with single-cell sequencing and other assays that might or might not be relevant. So typically, you go through, you collect your samples, you run them in the wet lab, you get your FASTA or FASTQ files, and you process them. Now everybody's doing MiSeq, or let's say even HiSeq. You run it through, let's say, for example, the QIIME2 pipeline, which strings together all these different tools for creating an abundance table. Traditionally, QIIME2 in particular uses a different method. They use Data-2 by default. But traditionally, you would have OTU clusters. So you would use the read similarity from each of the different samples. So for example, here we have 3 different individuals where they have red, blue, and green bacteria. And based off of a particular representative read, so one of those red reads, you can calculate what the distance is from from that read to all the other reads. And you define, usually it's at the 97% similarity, you define a cluster. So if you look at those little dotted circles to the right, you define a cluster of what defines an OTU, or an operational taxonomic unit. You then use these, and I won't go into the reasoning, a lot of it is for sequencing artifacts and technical noise, but you take these representative sequencing centers and you map them against a 16S database for doing annotation. Or you can do your analyses at the OTU level. So for example, back to this toy representation, we have reads from each of the 3 samples. And it happened to be that we had three reads that were red. These were from bacteria red that were present in sample 1. You see the first column, the first row. In the second sample, in second patient, we had 4 reads represented, 4 red bacteria. And in the third patient, we happen to have 5 red reads. And so you then look and you count up how many red reads you have in each sample, and you build this matrix, really. This matrix of OTUs, of red, blue, green, A, B, C, OTUs, so along the rows, by samples along the columns. And so the final output really is where we have, ideally, thousands of samples along the columns, right? And one can imagine you have multiple groups, right? You might have a healthy and a diseased group where you have maybe a couple hundred, ideally, or maybe you, let's say you even have just 10. You have 10 samples in group 1 and 10 samples in group 2. And you have OTUs, if you're talking about 16S, we're talking about thousands of OTUs. Or if you're using Dada-2, new pipelines, you might even just have hundreds of operational taxonomic units along the rows. And each value within that matrix represents the number of reads within a particular OTU that came from a particular individual. Apologies if I'm repeating myself, but this is probably the most important point to take home. That this is the starting point for many of the analyses that I do. That I then use R, and I develop R packages for analyzing this type of data. And in particular, I'm interested in, I'm going to present differential abundance testing. And so what that means is where we take a particular OTU or a particular species, if we aggregate up, or a particular genus, and we do a comparison of the abundance of the bacteria in group 1, and we compare it to group 2. So you can imagine the hypothesis test being, is the average abundance in group 1 equal to the average abundance in group 2? Is E. coli more abundant in healthy or in diseased individuals? So that's a little bit on the background for this. And this is a picture that I took from a slide from a prominent researcher in the field. And there's been a lot of hype about the microbiome right now, and some of it is much of it is justified, some of it is not. But everybody's talking about how the dysbiosis of gut microbiota is related to inflammatory bowel disease, rheumatoid arthritis, potentially depression, MS, asthma, colon cancer, all these other different issues. But people tend to forget that it's directly linked to infectious disease. And what I'm going to talk about is one of the earlier studies that I was very fortunate enough to work on, and that's diarrhea. Diarrhea, there's a directly understood and potentially even obvious connection to the microbiome, right? It's often a particular bacteria causing the disease in the gut. And understanding the dynamics of that gut bacteria, that's a causal agent for diarrhea, for this inflammation, is just a great testing ground for understanding the microbial communities in whole. So I was recently just at UCLA, and diarrhea in the Western world, you often think about it as just being a nuisance, but it's much more than a nuisance in the majority of the world. And there are approximately 800,000 deaths from diarrhea. I was just at UCLA the other day, and it's about 18 times the population of the entire campus. But 800,000 deaths a year, and it's just something that if we have a better understanding, potentially we could do intervention, or we can do better testing or testing of alternate bacteria. And so that was a lot of the motivation behind this. Approximately 1 in 5 children under the age of 2, they happen to suffer from an episode of moderate to severe diarrhea. And these children are really 8.5 times more likely to die within 2 months of having diarrheal disease. There's a relationship between growth and how growth is stunted compared to peers over the same 2-month period. So it really has a huge impact on the development of these children. Diarrhea, we have characterized diarrhea, and we know that there are viral agents, bacterial agents, and eukaryotic agents. Using 16S, it's only possible to characterize really the bacterial. But until now, until this study, it had been known that Shigella is probably the largest of the different diarrheal causal agents. I think it causes about 20%. But Shigella, Salmonella, Campylobacter, Jejuni, Aeromonas, Vibrio, cholera, and then various diarrheogenic E. coli like ETEC are known to cause diarrhea. So there are many known causal bacteria, but what we did as part of the global enteric multi-center study, there were a few, so this was a large Gates Foundation funded project across 7 different sites. There were a number of sites, 4 of them, the Gambia, Mali, Kenya, and Bangladesh, that really we had a molecular sub-study. We collected stool samples for the 16S targeted amplicon sequencing. And we decided to assay these samples. At this point, we have about 3,500 patients. So these are children, 3,500, but we reported a couple of years ago on the first 1,000. And so you can look forward to the follow-up study on the remaining 3,500. But so we published this study in Genome Biology about diarrhea in young children from low-income countries that really leads to large-scale alterations in intestinal microbiota. Back then, we used the unfortunately gone 454 flex sequencing technology of the V1, V2 hypervariable regions. It's important to note that we chose to use V1, V2 as opposed to V4, which is standard with the Illumina kit, primarily because we did not care about Bacteroides, and we were focused on pathogen detection, and the V1-2 we felt had a better representation, or we would be better able to speciate and identify particular pathogens that we wouldn't with V4. And this was a cross-sectional study where we had 1,000 stool samples from children under the age of 5, where half of them had moderate to severe diarrhea, and the controls were defined. We tried matching to the best of our ability within the same site, but the almost matched controls were children with no diarrhea within the last 7 days. So there were a number of questions that we really wanted to answer with this. How does composition of the microbiota differ by country and by age? How does the composition and co-occurrence differ in children with diarrhea? And the main question that we had to develop new approaches to analyze the data were if we could identify bacterial species that are associated with diarrhea. And that's where we had to develop novel statistical methods and normalization approaches for detecting differentially abundant species. So I'm going to spill the beans, spoil it a lot. Basically, the question as to whether or not we can identify bacterial species, and yes, we can. We can apply presence-absence statistics. So using ones and zeros, defining some threshold for presence and absence. We can, for example, do Fisher's exact test. That's one approach. Or you build a contingency table and you say, what are the odds of this particular bacteria or OTU or species or genus being in cases compared to controls. Additionally, we developed abundance-dependent statistics to test for if there's a difference in the abundance, right? Maybe the presence-absence doesn't matter. Maybe there's a ubiquitously present bacteria, but if it's at a particularly high abundance, then it triggers this cascade of inflammation. So we found a number of OTUs that were positively associated with diarrhea. According to all the different statistical tests that we measured, we found Granulocytella, which is known to cause swine dysentery, and we found some Streptococcus mitis and pneumoniae groups that were really previously uncharacterized and unassociated, had not previously been associated with diarrhea. And we also recovered in abundance-dependent methods, Campylobacter jejuni and Escherichia shigella. I should point out that Escherichia and Shigella, according to the 16S, you can't really tell the difference, right? Because they're the same. So, and as a little teaser, I'm working on a study right now where we're trying to compare neonatal sepsis patients that develop hydrocephalus. And there's an ongoing project with Dr. Schiff, Stephen Schiff, comparing samples from patients, neonates from Mbale and Imbarara in Uganda. So some exciting new work that's coming out that we're using 16S sequencing for as well. And so, yeah. So essentially, when we first started analyzing this data, we applied the very popular statistical methods. We didn't want to reinvent the wheel, basically. We applied methods that had been used in microarrays and for RNA-seq data. We applied EdgeR, and everything was significant. Applied DE-seq, and nothing was significant. There wasn't enough power. And that's because when you're dealing with these large sample sizes, there are a lot of distributional characteristics that are innate to the data that just really don't convey the characteristics of the data, right? So we needed to develop novel approaches, both normalization, so I'll go over normalization, and the actual testing framework for testing these different, whether a bacteria is more abundant in one group versus another, and estimating more robust fold change estimates. And that's because we're still in the wild, wild west when it comes to the microbiome, right? Slowly now, over the last decade, I've seen it shift. We're no longer just exploring, we're actually looking at mechanistic patterns. And so I don't know if I'll be able to use this, what I have thought is an amazing slide, but I don't know if I'll be able to use this slide much longer. But for now, I think we still are in the wild, wild west a bit. And that's partly because features being the taxa, the bacteria, they're unknown a priori. We don't, often you're doing this OTU clustering, right? It's based off of read similarity. And these bacteria are not necessarily within the databases, okay? And there are other issues, like I said before, that particular OTU matrix that I analyzed with the Gates Foundation, you can download it from Bioconductor right now, and it's 98% of the cells of the values in that matrix are 0. And that's highly representative of a large assayed, many sample assayed microbiome experiment, stool experiment. And so we still want to get meaning out of this table, but with 98% of the matrix being 0, how informative can that be? There's preferential sequencing. Certain bacteria, it appears, really just happen to go through the PCR amplification process better and are then sequenced preferentially. There are potential subgroups that you really want to control for. So you want to appropriately model with your favorite statistician, right? You want to collaborate with your favorite statistician, ideally, help them earn a living, and control for sites, gender, countries, so on and so forth. And because of this, we have a large number of features, and so you always want to control for multiple testing and account for your type 1 error. So first I'll go into the normalization aspect and the normalization approach that we developed. On the left is a cartoon image of the count matrix representing the rows being genes, and where this is RNA-seq data. And the columns are representative of the various samples. On the right is a representative cartoon of metagenomic data, where the rows happen to be the OTUs, right? And the columns are still the samples. And the green shades, I'm not sure if it comes through for everybody, but the green shade represents a positive value, so something greater than 0. And you'll see on the RNA-seq, RNA-seq data is definitely more sparse. There are more zeros than this cartoon says. I'd say maybe 30%, depending on the experiment, are zeros. But in metagenomics, it really is that there's a huge difference between these. And oftentimes I'll see 90% zeros or higher in metagenomics. But so what RNA-seq data, what one approach did in particular, there's a number of different normalization methods. One approach, so I would define normalization in 2 ways. There's either scaling normalization, that's where you divide by a single value, or there's global transformation. So an example of scaling value normalization is where you divide by the column sums, so you would get proportions. So you divide by the library sizes, okay? Other approaches are and that would give you the relative abundance. And everybody in the microbiome is reporting relative abundance for different communities, and it's important to do that. Other normalization methods make different technological and biological assumptions. For example, in microarrays, it's very popular to do quantile normalization. And that's where the assumption is that the count distributions or the distributions for each sample are meant to be similar, right? But that they're only different due to technical noise. And so you force all of the distributions to be the same. So that's an example of global normalization. There are a number of approaches. There's TMM, which is used by EdgeR. There's RLE, used by DESeq2. And then there's also library size for proportions. But Sandrine Dudois' group over at Berkeley highlighted with a spike-in experiment a while back how in RNA-seq data using library size proportions was invalid, and because it led to greater false positives and greater false negatives. And instead, what they observed was that the low to medium quantiles, so the low to medium quantiles of the count distribution behaved similarly across samples, and that it was really at the tails of the distribution, the really high abundant genes that destroyed the characteristics that that varied across different samples. And so with that idea, what they decided was to scale each sample by the 75th quantile. And in that way, you're controlling for all the different values. You're controlling for the technical variability, the technical variation in sequencing for each of the samples. So here on the left, we have the original matrix. And what they would do is they would calculate for each individual sample, what is the 75th quantile of that sample's count distribution. And that particular value, the dark shade of green, would become the normalization factor used for scaling, followed by log transformation to be the normalization constant. Alternatively, we observed similar things in our data, that the low to mid quantiles of the count distributions, of the positive count distributions, behaved similarly across samples. But it was really the tails. It's these particular bacteria that are very, very abundant after sequencing that are absorbing and probably getting preferentially sequenced, that are getting all the sequencing going on, that are really biasing the proportions. So if you were to use proportions, right, you might downscale certain bacteria that are equally abundant across samples, but because there was one particular, a few samples that got a lot more sequencing, they were, and the most abundant bacteria were preferentially sequenced, you're going to get proportions that are not comparable and not representative. So instead, we decided to follow Sandrine Dudois' group, and we calculated, we felt that a 75th quantile, it wasn't always the appropriate value. So we decided to use a data-driven approach to determine at what quantile do most of the samples deviate from the reference, being the reference representative of the entire data set. So at what point do the quantiles really shoot off from the average of the entire data set? And also, instead of just using that particular quantile value within a sample, scaling and summing up all the values from the low to mid quantiles up until that particular quantile of the sample, and using that sum as the scaling normalization approach. So, and that's represented here, where you have the original on the left. You have the original matrix. You sort the values, the positive values, and you calculate the quantile, and you sum up the values up until that quantile for each of the samples. What happened was we took, as a representation of why this normalization approach works, we applied it to an older data set by Peter Turnbull, where they had two sets of mice, fed the same diet for a couple of weeks, and then one group of mice, they introduced a new low-fat polysaccharide-rich diet. So, they went from a low-fat polysaccharide-rich diet to a Western, I like to call it the McDonald's diet. And here, each of these plots, A, B, C, D, we see different approaches for normalization. And in the A, the one highlighted, is my particular approach called cumulative sum scaling normalization, where you see that we're better controlling the technical variation and highlighting the biological variability there. You can also download this particular data set from Bioconductor. So whereas with DE-Seq's RLE normalization and with TMM and total sum, you get some separation, but it's not as representative, and it's not controlling the technical variability as well. Additionally, we made use of linear discriminant analysis, and we were better able to predict whether or not a particular mouse came from the Western cohort or from the low-fat polysaccharide-rich diet. So I'm going to shift gears a little bit from normalization, though there's been lots going back and forth about normalization. especially surrounding pseudo counts. And my argument about that is if you're going to include a pseudo count, I would recommend using 1. So a pseudo count is where you take the count matrix and you add 1 to everything, followed by a log transformation. Because if you're going to be doing PCA analysis, so that's what's represented here. These are PCA or multidimensional scaling plots. They allow you to see the most variable components in a data set. Which here you see the greatest variability happens to be biology. If you're going to add less than 1, what's happening is once you log transform, you're mapping a 0 value to a negative, and so the greatest variability is going to be between what is present and what is not. All right, so at this point, I'm going to shift to the hypothesis testing of differential abundance, where we're looking at, for example, just OTU3, and we're looking at if OTU3 is more abundant in group 1 or in group 2. I'm going to remind everyone how we're looking at what is the differential abundance. We want to make use of that linear relationship between sequencing depth and the number of detected features. So we want to make use of this linear trend, and it's really a Bernoulli process, right? Bacteria is present or it's not, and it's related to how much sequencing was output. So what we did, at the time, we decided to make use of a zero-inflated Gaussian mixture model. Essentially what that is we apply a sample, we estimate the probability that a particular value or a particular sample's OTU comes from either a detection distribution, so a spike mass at 0. If you look at that little distributional cartoon on the far right, there's like a little, I don't know, lamppost, right? That is like the technical technical distribution. It's a detection distribution for zeros. This is for 102, right? And so we estimate what's the probability that a 0 in a sample, given a sample's depth of coverage or sequencing depth, what's the probability that it came from technical noise? So somebody kicked the sequencer and the sequencer decided not to spit out a lot of information, or that it was truly present and it comes from this particular biological distribution, which at the time, we simplified and made use of Gaussian. So we modeled all the data as coming from the zero-inflated Gaussian mixture model, and we make use of the probabilities that a 0 belongs to the biological distribution to get better, more robust fold change and variance estimates for the differential abundance testing. Here's a little cartoon for those that are more quantitatively inclined. I'm not going to go over it, but essentially in the dark green, you have your observations. So Yij are going to be your observed counts. Kj is the status, let's say case control. And Sj is the sequencing depth. So using those three, we can model what are two distributions. So our two distributions are the spike mass at 0, the two light yellows. So Y0, the spike mass at 0, and Y1, which we model as Gaussian. And the delta IJs, so the gray, we can estimate using our logistic, using a logistic and an expectation maximization algorithm. The details are in the paper. What the posterior probabilities are for a 0 belonging to one or the other of the distributions. And then the light grays on the far right, we can estimate those fold change estimates, variance, and baseline estimates for each OTU. For a graphical explanation, here, if you look at one particular row, I'm presenting Granulicatella paradisians from subgingival plaque and tongue dorsum. On the left, we have kernel density estimates for if you look at the red, it's subgingival plaque. If you look at the blue, we have tongue. So, and we see a difference between the two groups, right? This particular bacteria is more abundant in the tongue. So without accounting for the sequencing depth on the left, we see that there's a large difference. However, a lot of the samples that from the subgingival plaque happen to be patients or individuals. These weren't patients. This is HMP. These were all healthy volunteers. These individuals, it just so happened that a lot of these samples had lower sequencing depths. Now, and we believe that in part was due to technology, not necessarily biological. Once you account for the zeros, once you account for the sequencing depth, you see that the kernel density shifts, right? Those samples are estimated lower or down-weighted, and we see that there still is a difference between the subgingival plaque and the tongue dorsum, but that difference is more attenuated. There's a more moderate difference between the two groups. So it is still there, but it gives us a more robust estimate of that fold change. And so we compared, here are Manhattan plots where every positive spike represents the abundance within a particular group, within a particular sample, sorry. For this Latropia oral clone, where on the left, we had 120 samples, so you'll see 120 lines there of the abundance for this Latropia in subgingival plaque individuals versus tongue individuals. And so we see that Latropia was highly present and highly abundant in the subgingival plaque, whereas in comparison, it was not present at all in tongue. And so when we did a particular analysis with, for example, LabRoots at the time, because one approach is to do all these multiple comparisons, we included site information, and there is a batch effect. Those dashed dotted lines in the figures represent the sequencing center. So you can actually see in the subgingival plaque, one of the sequencing centers didn't see any of practically any Latropia in subgingival plaque. But in either case, if you include that variable in LabRoots, for example, you don't observe any differential abundance for this particular organism. But you do when applying Metagenome Seek, our approach that encompasses the normalization and differential abundance analysis. Coming full circle, we were able really to also highlight Granulicatella, which I already said is known to cause swine dysentery, and we were able to find other species as well that are novelly associated and are being tested with TI-84 cell and epithelial cell lines and other cell lines, and in the animals, if the associations are causal. So now you can look forward also to a whole new framework. There were issues with having too high of a false discovery rate, so too much power, and we want to curtail the false positive rate. And so we restructured it under the hood, and now within Metagenome Seek, there's actually a fit feature model function to allow for a zero-inflated log normal. And this is different from a zero-inflated Gaussian distribution that really allows for much faster computation because there's no need for an expectation maximization algorithm. And it really, I don't know, I think it's superior to the older approach. But all these approaches and methods are within Bioconductor, in a package called Metagenome Seek that really encompasses the full statistical framework that I've highlighted here and the utility in doing some quick and dirty analyses. PhyloSeq, a very popular bioconductor package for microbiome data, also feeds in, and there are wrappers to creating Metagenome Seek objects to do these different analyses. Though I recommend Metagenome Seek more for larger studies. There's a large infrastructure and a set of data packages. For example, this whole data set that I described is available in a package called MSD16S. There's one where we challenged individuals with enterotoxigenic Escherichia called ETEC16S. But revolving around this Metagenome Seek package, there are a number of visualization and infrastructural components and packages that you can be on the lookout for. Metagenome Features was just finished, and we have a paper in submission that will allow for exchanging of annotation of 16S data between green genes, SILVA, RDP. There's formatting packages, one of them being biome format that will incorporate, load into R biome files that I worked with the PhyloSeq author, Joey McMurdy, on. And then there are visualization tools, one of them being MetaVis that came out, I think, a month ago in nucleic acids, led really by Justin Wagner. Oh, I want to do a quick shout out. Metagenome Features was built by Nate Olson in Hector Bravo's lab. Did a great job on that and other developments. And then MetaVis really led by Justin Wagner. This really cool interactive and statistical and visual analysis of metagenomic data that I hope LabRoots would invite Justin for a talk going forward. Really, that has, I think, 70,000 samples loaded already into the interactive visualization browser. Essentially, go to the links on this webpage, and you can see and play around with some of the data. In the article itself, there are a couple of figures that are static, and you have the ability to go and directly get linked to them. So as a roundup, I just want to highlight that whenever you're doing a microbiome analysis, it's because of the sparsity, because of all the zeros there. Depending on the sequencing site and other assay characteristics, it's very easy to find batch and center effects solely due to what's missing. Here, I'm presenting fossa from the Human Microbiome Project, and I can show from HMP2 and from other more recent studies as well. But there's a difference in for the same particular body site that we don't have using this particular data set a way of characterizing whether it's due to the individual or due to sequencing center, but I can tell you it's due to sequencing center, where we see that there's a very large difference in the fossa. Okay, here not necessarily because of the overall proportion of positive OTUs, but because of which OTUs were present in the WashU sequencing center. We see a large distribution. Oh, this is another PCA or MDS. You can think of it as a PCOA plot if you're visualizing data, if you know what that is. But it's another way we see these huge batch effects. And so I highly recommend maybe even instead of coloring by sequencing center or batch, your PCA plots, but maybe by shades of the proportion of positive OTUs within a particular sample. For example, here's a more striking difference, where in gingival epithelium, you can see differences in the proportion of OTUs by sequencing center. So in the top left, we see the proportion of positive OTUs in box plot format, where JCVI in green is at the bottom end, and WashU in purple is at the top end. And when you do your PCA, you see you can really see where the two sites diverge. They're at opposite ends of the PCA plot in the first coordinate. At the bottom left, we also see that PC principal components are highly correlated with sparsity and obvious sequencing batch. So just a little cautionary tale and some other pieces that there's ongoing research for. And so I really just want to take this time to acknowledge and thank, all the people that have worked on this and that I'm working with at the University of Maryland, Mihai Pop, Hector Carrara-Bravo, Colin Stein. At Penn State, my collaborators on the Uganda neonatal sepsis project that I think some very exciting work will come through, and others.
Thank you, Dr. Paulson, for that informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question drop-down box located on the far left of your presentation window. Type your question into the box that appears on your screen and click the Send button. We'll answer as many of your questions as we have time for. So let's get started. Our first question is, where can I find Metagenome Seek?
So Metagenome Seek is available at bioconductor.org. You just search, there's a search toolbar at the top, and you just type in Metagenome Seek, and it should be the first link. It should be easily findable, even through Google.
Great, thank you. How do I install Metagenome Seek?
So on the page, when you go to Bioconductor, go to the Bioconductor website for Metagenome Seek. There's a little gray box that's going to highlight in R the code necessary for installing. So it says installation. It says to install this package, start R and enter source BioC Lite Metagenome Seek. So copy and paste those particular lines of code, and then you'll get it'll start installing, and it might take a few minutes.
Okay, we have time for one more question, and it is there a user guide for Metagenome Seek?
So every particular bioconductor package comes with a vignette, and there are 2 vignettes associated with this. So there's browse vignettes as a function with a capital V, and then you type in the package name. In particular, this would be Metagenome Seek, and you'll find detailed documentation on various functions and guides for, and not just for installation, but also for analysis pipeline.
I would like to once again thank Dr. Paulson for his presentation. I would also like to thank LabRoots for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through August 10th, 2018. You will receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now. Thank you for joining us. We hope to see you again soon. Goodbye.